Hello, my name is Rotem Karni and I work in the Institute for Medical Research, Israel, Canada, in the Hebrew University. And we work on cancer. And when we talk on cancer, we talk on three things: detection, treatment, and cure. And when saying this, we are basically losing the war on cancer in all of these fields. So detection, we don't have good enough detection markers to detect cancer before it starts, and then we can operate patients. We don't have good enough treatments, as all of you are probably aware of. And eventually, of course, we want to reach the cure and to cure people from cancer. So what we are doing in the lab to solve this problem? We are working on genes which are called splicing factors. These genes are commander genes. They tell other enzymes in the cell or in the body how to manufacture new proteins in the cells. But sometimes these commander genes turn bad. And when I'm saying turning bad, I'm saying that something happens to these genes. For example, in many cancers, these genes are overproduced in very high amounts, and then they start to give conflicting orders, and this is very deleterious and can cause problems in the cell and eventually lead to cancer development. Recently, we identified such a gene, which is a commander gene, which is really overproduced in brain cancers, and we even show that it can cause brain cancer. We recently published these findings in the journal Cancer Research of the American Association of Cancer Research. And in this publication, we found that this commander genes is really upregulated in brain cancer, produced in high amounts, and it can predict even the survival of brain cancer patients. But more than this, this gene actually can cause brain cancer. So if we take normal cells and we put these genes into these cells, we can turn them into cancer cells. So this is a really important gene that controls the development of brain cancer. And we show that if we can inhibit this gene, if we can block the production of this gene, we can actually block the development of brain cancer, at least in animals. So our next step is to try to develop treatments that will use this new target that we identified as a new type of therapy for brain cancer. And the idea is to block the manufacturing of this factor, of this gene, and by this to inhibit the development of brain cancer. And we are trying to do it with several companies and to develop our own technology to do it. And we hope that eventually it will lead to the development of a new cure for brain cancer. So how do we study the activity and the involvement of this gene in the development of cancer? What we do in the lab? So first of all, we manipulate these genes, either overproduce them or silence them in cells in the dish. And we can do it genetically or pharmacologically. And then we can do it also in animals. So we treat the animals and we inject cells, and these cells bear tumors. And you can see here, for example, a lung full of tumors. And when we block the production of this splicing factor, of this gene that we identified, we can actually clear the cells from all the tumors, basically inhibiting the cancer. And eventually, of course, we want to move to humans and to cure humans. So we start from cells, we move to animals, and eventually we want to try to do it in humans. It's a long way, and it's a long journey, and this journey needs a lot of funding in the way to find people, and to pay for people, to pay for all the reagents that we use, the animals, and all the facilities that we use. And for this, we really want your help in advancing in this progress towards the cure of cancer.